Good evening, friends and colleagues, ladies and gentlemen. The IMS public lecture is an outreach activity of the NUS Institute for Mathematical Sciences. This evening, we are honored to have Professor Samuel Safran as a speaker. Professor Safran has been a professor in the Department of Materials and Interfaces of the Weizmann Institute since 1990. He has also served as Vice President of the Weizmann Institute and Dean of the graduate schools. He currently holds the Fern and Manfred Steinfeld Professorial Chair. From 1980 to 1990, that is before he joined the Weizmann Institute, he was at the Exxon Corporate Research Labs, where he worked on the theory of soft matter with a focus on the structure and phase behavior of oil, water, surfactant dispersions. His recent research interests have extended soft matter concepts to treat biological membranes and cells. Professor Safran is the author of a graduate text on statistical thermodynamics of surfaces, interfaces, and membranes, and the recipient of the Pierre-Gilles Lejeune Prize Lecture Award, the Bella Lectureship Award, and the Lectureship of the Chemical Society of Japan. It is now my great pleasure to invite Professor Safran to deliver his public lecture, Order and Rigidity Sensing by Biological Cells. Professor Safran.
Thank you for that introduction. That gave me a lot of honor. I hope I'll fulfill the honor by giving a good lecture that you will enjoy and also understand. That's really the main thing. It's a pleasure to be here. It's my fourth trip to Singapore. I have usually I was at the material science meeting in June, and twice have visited the Institute for Mechanobiology, which is on this campus, and that is an interdisciplinary institute with mostly biologists, but also physicists and chemists, material scientists. Daniel Rivline, my colleague, is here. He's giving some lectures there as well. I don't know if they have mathematicians, Daniel. No, I don't think so. But maybe after speaking to this audience, you will be able to make connections with the Institute for Mechanobiology. But I'll tell you a little bit more about the physical properties of biological cells. And we'll talk about the fact that the physics of the biological cell is also important for its function. And that's something that experimental biologists working together with material scientists, physicists, and chemists have been trying to understand. And what as a theorist, we are trying to provide theoretical insight to that. I'm going to show you a lot of pictures tonight because it's just a general talk. I'll also have some equations. Those of you who come from biology and don't want to look at the equations, you can close your eyes. And those of you who like the equations, well, you can still look at the pictures. But you'll see that there is a theory behind all this, and if somebody's interested in details, we can give you references. The main point of the talk is to understand that cells respond to various forces, to external forces. And they respond in such a way not just to change the way a piece of dead material would change if I stretch it or if I shear it, but they also respond by changing their function. I'm going to show you several examples where you exert a force on a cell, either because you pull on it, or you put it on a surface with a certain property, or because the surface is curved. And when you know that when you curve something, you're really bending it, cells respond differently. And we're going to talk a little bit how the internal order in a cell depends on what's going on in its environment. I'll give credit to the people that I work with, the people in my group. There are a string of many, many postdocs over the last 10 years or so. Ulrich Schwartz is now professor of Heidelberg. Ruby Day is back in India. Yair Shokef is a young professor now at Tel Aviv. Asi Zemel, a young professor at Hebrew University. Benjamin Friedrich has gone back to Dresden. So really with a string of postdocs we've developed this, and we've benefited very much from experimental interactions with a group of Stuttgart, a group of Joachim Spatz. Ralph Kemmerer is the group member who's a scientist in that group who's done many experiments. I think he's supposed to be at the Mechanobiology Institute. No, he didn't come. Okay, the group of Rudi Merkel is another physics group doing experiments on physical properties of cells in Jülich. And today I'll also tell you about a theory we've been doing in conjunction with experiments done in the group of Dennis Disher, University of Pennsylvania. And our work in this area really started as an internal collaboration with Weizmann, with Sasha Bershadsky, Ben Geiger, and Leah Duffy. Okay, I'm going to motivate this by something that really motivated many, many people in this field, which is that cells respond to external mechanical cues. But they don't just respond mechanically the way a piece of paper or a piece of plastic or a piece of metal would respond if you stretch it or do something. What happens is that the cell function changes. Cell function is sensitive to its mechanical environment. And the group of Dennis Disher, which is an interdisciplinary group, Dennis comes from polymer science, material science, physics. They have biologists, physicists, and material science in their group. They're an experimental group. Did a very interesting experiment published in 2006. He took stem cells. Stem cells are cells that come from the bone marrow or from the umbilical cord, and they're cells that can develop into any kind of cell. So those are the cells that medicine wants to use as the ultimate way for the body to heal itself. And what he found, now usually biologists that deal with stem cells put them in different biochemical soups, so some complicated chemistry that the stem cell is in, and they try to make a kidney, to make a liver, to make heart muscle. What Dennis did is he kept the biochemical soup the same, didn't change it, but he changed the physical conditions on which the cell lived. In particular, he put the cells on a plastic substrate, and he changed the rigidity of that substrate, how hard it is to stretch the substrate. And what he found after 96 hours is that cells that were on the softest substrates developed into nerve and brain cells. On the hardest substrates, they developed into bones. And on intermediate substrates, they developed into muscles. Now, those are pictures of the characteristic, what's called phenotype, or cell shape. But also the genotype, the biochemical gene expression, the proteins that the cell makes, its biochemical function, was also affected by this, as this plot shows. For instance, this green curve here are markers indicative of muscle cells, and they only were expressed by the cell only produced them in a certain range of substrate rigidity. The same for brain markers in this regime, the muscle markers in this regime. So this was a very fascinating result to biologists, bioengineers, and even theoretical physicists as well. And this is a motivation. We're not going to explain this in great detail. I'll tell you some ideas we have about it. A similar motivation is called the field of tissue engineering, where people want to make artificial organs. And these are experiments that were published a while ago in PNAS by this group. That was, again, a physicist, Gabor Forgas, and Vladimir Mironov is an actual medical doctor who does research. An interesting combination. They put cells on a gel. They did it with an inkjet printer. They put the cells instead of ink and the gel is instead of paper. And they watch how these cells aggregate. They interact with each other through the gel. They feel the gel. They're pulling and pushing on the gel. And they interact with each other. And in this picture, they think they're making blood vessels out of it. The dream is, of course, to have any kind of cell just get together and make an organ that you would like. Right now it's still a dream, but there are a lot of science issues behind us that are related to my talk. I'll show you another movie about the mechanical response of cells. These are experiments of Ralph Kemmerer and a group of Spatz in Stuttgart. These are cells, they're connective tissue cells like you have in your ligaments, and they're put on a plastic gel, and that gel is stretched in this direction, and it's stretched at 1 hertz, just about like this, with an amplitude of 8%. And unfortunately the movie didn't play, so I'll have to start it up from here, and you'll see what happens. The cells respond to the direction of stretch, and they respond by going perpendicular. The stretch is horizontal, and the cells align nearly perpendicular to the direction of stretch. There's some distribution in angles, but it's a very striking phenomenon. Now they stop the stretch. And what do you think happens? The cells don't stay where they were. They go back to their random positions. So they started out in random positions. You stretch, and you stretch for quite a few hours till they finally align nearly perpendicular. You stop stretching. They go back to their random position. So this means that they're feeling the physics, and they're feeling the physics in a somewhat reversible manner. Other examples of mechanical response I'll show you in a minute, but let me just give you a hint why a cell responds mechanically. Now, many of you know cells have a cell membrane, and there are sort of a lot of chemicals inside. That would be sort of a mathematician's or even physicist's picture, which is true. A cell is 70% water, but it's not a bag of fluid. Namely, cells have characteristic shapes. And we know that fluids don't care about shapes. Fluids only care about their total area on their interface. They don't care about their shape. To care about their shape, you have to be a solid. You have to be connected. And the reason cells care about their shape is internally there's a gel of a long chain molecule called actin. It's a protein. The details on the level of the molecule aren't important, but it makes long chains, and those long chains intertwine. And they're also cross-linked. I drew a sketch over here of what I mean. These are the long chain molecules. They're called polymers. Actin is a molecule that makes very, very long chains, up to a micron long, even longer. And there are cross-links that are these black dots. You can't see them here, but they're molecules that join the chains to each other. And in a sense, this is a solid. And not just in a sense, strictly speaking, it's a solid. It's not a crystal like a semiconductor or metal. It's not a periodic arrangement of atoms. These gels are very, very random, as you see here, but this responds to stretch and, more importantly, to changes in shape, to shear. If I hold the bottom constant and I push the top, this will change its shape in the same way that a rubbery or solid material will change. Why does it do that? Because of these crosslinks. That's how this is different from a fluid. These crosslinks hold the chains at these places. They can be stretched. If the crosslinks stretch, this chain stretch, and it costs some energy. But they cannot, the crosslinks don't diffuse the way the molecules in the fluid diffuse. They stay more or less at these positions with some deformations. And we'll talk more about deformations in a mathematical sense. And this gel is only one part of what's called the cytoskeleton of a cell, and the cytoskeleton is a network, not only actin, but there are other molecules as well. And that's the reason, even though a cell is 70% water, the cell has structural integrity. The cell knows about its shape. If you change its shape, it will respond and it will move back to its shape. So you put a force on it. While you're forcing it, it will deform, just like a piece of solid material. But after you remove the force, very often the cell will come back to its shape, like a piece of solid material. But it's a very, very weak solid. The distance between crosslinks here might only be 100 or 80 nanometers. And in a solid, or typical in a metal, the atoms are angstroms apart. So this is a very weak solid because you're holding it together only every 100 nanometers, not every 5 angstroms. And so the forces here are much, much weaker, and the elasticity of this gel is 6 orders of magnitude lower than the elasticity of a typical solid material. One of my colleagues, Jeff Fredberg at Harvard, says, if you want to understand what a cell feels like, it doesn't feel like wood, like a piece of metal. If you would take your finger and mush a cell like this, it would feel like toothpaste. But it's still a solid. It's still a piece of toothpaste, has a shape. It doesn't flow away like a liquid. Okay, now there's one other thing we have to know about cells. You have this gel, so it acts like a jelly, like the jelly that you eat, or other kinds of gels. It's a weak kind of solid, as I've indicated here, because these crosslinks are far. But there's something else. Cells are alive, and cells eat, or we eat, or animals eat, or everything alive eats, burns energy. And when it burns, one of the things that cells do when they burn energy is they have molecules in them called molecular motors. And this motor is called myosin. And what this motor is doing, it's a little frustrated. It has 2 parts. This one is a motor, and I won't go into the molecular details. It's trying to move on the actin in this direction. And this one's a motor trying to move on the actin in this direction. Remember, the actin are these long cross-linked molecules The myosin, the green, are these motors, and they're trying to move. Well, if this tries to move here, and this tries to move here, and they're connected by something rigid, they can't really walk because they're each trying to move in the opposite direction. Well, what they do is they exert force on these actin chains. So the cell is a gel, and inside the gel are little molecules that are tensing the gel. They're always pulling on the gel. And indeed, if you put a cell on a piece of plastic and don't stretch it or do anything, the cell will crinkle, will wrinkle the piece of plastic. Not as much as my hand is wrinkling this piece of plastic, but it's actually like little muscles in the cell that pull on the piece of plastic. So since there's a theoretical audience, we'll take just a minute to talk about pulling and forces, just to remind you of some definitions. But I said it's not necessary to follow any of the equations to get the idea of the talk. All right, if you have a solid material, as I said, connected either because of atoms that are interacting, or these cross links that cannot diffuse, they connect these molecules, they can be stretched, they can be deformed, but they don't diffuse all around. That solid has elasticity, and you can deform the solid. You move the point P and the point P0, some other distance, P prime, P0 prime, and the displacement is the distance that you relatively move these 2 points, that you moved one of the points away from its equilibrium position. And the strain, which is a tensor, and it's a standard definition, I've written it here only in words, it's the change in the relative cross-link distance. So it's not just the motion of one cross-link, because you could take all of them and just move the whole gel. That wouldn't cost any energy if I just translate the gel. But it means that I've relatively moved 2 of the cross-links, and I've stretched this chain. It's the relative motion that's important, and that's why there's a gradient. So there are strains that represent the relative motion of these cross-links in the gel. Another word we'll use is stress. We'll define that in a minute. And here's what I meant by shape change. You take a solid that was a rectangle, and you put a force on the top, you'll get a change of shape like this, and this is known as a shear of a solid. Of course, you know you could compress or stretch, but usually many gels, because there's water inside, are not very compressible. But you can shear them. You can change their shape. You hold this area fixed and put a force on this one. The other word we have to know is stress, and you could ignore the equations. Stress is the force per unit area applied to a given plane in a solid. What I wrote here, just for simple, it's the force per unit area in this gel if something makes these crosslinks pull apart. And what would that something be? That something is, in the case of the cells, these myosin motors. They're always pulling on this actin gel, and they're deforming the gel. And that causes, of course, I said they'll stretch this little piece of gel in between the crosslinks, but that costs energy, or it costs entropy. It costs some free energy to do that, to make that stretching. So just like in a regular solid, there's a force that's intrinsic to the material. If I pull these 2 crosslinks, this stores energy in it, and there's a restoring force that wants to go back to where it was. That's all the elasticity we'll need. Now let me continue a little bit with mechanobiology. As I said, if you put cells on gels, they deform the gel. And this was an experiment done at Weizmann by Natalie Balaban, Benny Geiger's group, together with Sasha Vershatsky and others. And my postdoc, Ulrich Schwartz, did the mathematical analysis of this. And they made a gel that had a pattern in it. They put a cell, here's the cell on it, and they could see that the pattern is deformed here, here, and here. From those deformations, you could ask, what were the forces that the cell exerted on the gel? In mathematics language, you have to solve the inverse problem. You don't know the force and predict the displacement of the gel. You know the displacement, you have to predict the force. And this was the answer. The blue arrows were the answer. The blue arrows are the forces that the cell exerts on the gel. So the cell is pulling on the gel, and it's pulling more or less in this direction here, and more or less in this direction here. The cell is glued to the gel by these little green areas that are called adhesion molecules, that are a type of glue that the cell makes to attach itself to its substrate. And the red are these actin fibers. Those are these long molecules that in this cell have arranged themselves in parallel long fibers, and that's part of the ordering we'll talk about. And as I said, you can solve this problem. They did the measurements. They found where the gel was deformed. It relaxes. If you get rid of this actin gel, then we'll get rid of the molecular motors. The gel is not the substrate, I'm sorry, the pattern substrate is not deformed, so relaxation here means you've gotten rid of the motors or disembodied the gel. But before the thing is deformed, from the deformation, you can solve this inverse problem where you know what the force is, and this G is Green's function. It comes from theory of elasticity. It's sort of a little bit like Coulomb's law in electrostatics. It goes like 1 over the distance between the forces. So the displacement at point R depends at the force at another point. And that makes a lot of sense. Because if I have a connected network, and I pull over here, because the whole network is connected, this point will be displaced, even if I pull far away. And that's the elasticity transmitting the forces through the elastic properties of this network. So cells on surfaces, even if you don't do anything to the surface, the cells pull on the surfaces and they deform them because of these molecular motors. Okay, now one picture of an entire cell is something that's been known in the mechanical literature, in the materials literature, in the physics literature as a formed dipole. In a sort of very average sense, the cell is pulling this way in this direction and this way in this direction. It's like two forces that are equal and opposite. You all know you have electrical dipoles, but this is a dipole of two forces that are equal, more or less equal and opposite. That those two forces are separated by distance L. You can define a dipole moment, which is the product of the distance times the force. It's a tensor, and we've written many papers about the theoretical properties of force dipoles as applied to cells. We'll meet dipoles in another minute in a different context, but this is an entire cell model of the force dipole. Now, as I showed you, cells respond to dynamical elastic stresses. I showed you the movie. Just to review, when you have a static stress, in some cases, but not all, cells will align parallel to the static stress. But exactly what happens if I stretch and hold is something that still is not so known. What's more well known is if I do a dynamical stretch. I stretch like this. You might ask yourself, why is he talking about dynamically stretching? What in the body could that be relevant for? What it's relevant for is very simple. All the cells in your blood vessels feel the blood pumping, and the heart is pumping, relaxing, at about 60 beats a minute. So at a frequency of 1 hertz, those cells are feeling the fluid, the blood, that's pulling on them or shearing them, that keeps changing dynamically. And that's why dynamical experiments are important. Also, you might think if you were making artificial tissues, you might use dynamical stretch or static stretch to process your tissue, to align your cells, like in the movie I showed you. So this is one kind of force that I've already shown you a movie of, showing that cells respond to dynamical stretches. There are other stresses that you can put on cells. All you have to do is put them on a curved substrate. I don't know how many of you have studied elasticity, but if you take a piece of paper, okay, and you bend it like this, it doesn't bend spontaneously. You have to exert a force. And the reason you have to exert a force is that when you bend it, the inside is compressed and the outer edge is expanded, and that costs energy. You're stretching, you're compressing, and that is bending. It's well known in elasticity. And when you put cells on curved substrates, they respond by putting their stress fibers. Remember, those stress fibers were the ordered long chain molecules that actin gel sometimes orders. I'm not sure I defined it too quickly here. I'll show you again. These are this actin gel, but instead of being a cross-linked gel that's all random, all these actin fibers, these are not individual polymers, but fibers, they've aligned parallel, they've ordered. But it's the same actin myosin gel is just ordered. And when they're ordered like this, they're known as stress fibers. Here, the stress fibers can align along the curvature axis of the cylinder, or they can be parallel to the curvature axis of the cylinder, and depending on the type of cell, smooth muscle cells or connective tissue cells, they go in different directions. So I'm not going to tell you why. We have some theoretical idea, and there are papers published by the group of Sasha Bershatsky a while ago on those experiments. I'm going to talk more about what happens within the cell. I've defined a force dipole. A force dipole are two forces that are equal and opposite and separated by some distance. Like an electrical dipole is a plus charge and a minus charge, separated by a distance. And that's what happens with this little piece of actin and myosin. You have the myosin pulling in two opposite directions. It's putting a force on these actin. And this little unit we can call a force dipole, and that's what these little pictures represent. Now within the cell, these actin-myosin dipoles can order. And in your muscle cells, they're very, very well ordered. They're ordered, these are fibroblasts, but it doesn't matter. They're ordered very much like this. They're all parallel, and all those dipoles are ordered together. But if you have a stem cell, like I was telling you about at the beginning, the stem cell is the baby cell. It doesn't know what it's going to be when it grows up. And its actomyosin, its little pieces, start out in the stem cell as little random pieces. I'll show you an experimental picture in a little while. And they don't know which way to order. If they're going to be a muscle cell, they will have to order all in the same direction, like those red fibers I showed you. If it's going to be another kind of cell, it's not so important to order. The muscle is very important to order. Other cells, it's less important. And I'm going to tell you a little bit about what's called pneumatic or orientational ordering of nascent stress fibers. That means the stress fibers are just being born. We're talking about a stem cell. The experiments we're talking about are stem cells. These stem cells have these little pieces of actomyosin, the gel, and the motor that pulls on the gel, but they're nascent. They haven't made long, long fibers that have been ordered. Instead, they're just little pieces that are isolated from each other. Yet, because they're connected in an elastic network, when one of those pieces pulls on the elastic network over here, so when this piece of actomyosin is being pulled like this dipole, because you have more elastic gel in here coming from the unordered actomyosin network and other molecules, this elastic network means that this one feels that this one is pulling. They interact with each other. This one pulls, and this one feels the pulling because there's a whole elastic network that's combining them. And the fascinating thing is that pulling can serve to order these dipoles from a state where they had no order, they were pointing in random directions, to a state where what's called they have pneumatic order. It means they're all pointing in the same direction. They can show more complex orderings as well, called symmetric order, and I'll just show you some pictures. And what's very interesting is that what was found experimentally, and we actually did some of this theory first, and then Dennis did the experiments, was that this ordering depended on how rigid the substrate was. So now we have two pieces of information. We have the original experiments I showed you on the stem cells, where the function of the cell, the biochemical function, the proteins the cell produces, the shape of the cell, all that depends on how rigid the substrate is. That was the second slide I showed. And now we have another function that is purely physical, or almost purely physical, which is how well ordered these little pieces of actomyosin are in stem cells. And this also depends on the rigidity of the substrate. So we have two pieces of information, one very complicated biochemical, one a little more simple, more physical. We'll see if we can combine them. This is, to use the language of the workshop in the Institute, it's a multi-scale problem. We have these little pieces of actin and myosin, things that are 10, 20 nanometers in size. We have the bigger stress fibers that are 100 nanometers in size, something like the distance between these crosslinks. And we also have the cellular level, we have the stress fibers, the adhesions, and the entire cell is a contractile element. So depending which level of detail we're looking at, we can look at the 10 to 20 nanometer, 100 nanometer, micron, or 50 micron scale, and there's interesting science in all of these areas. And not only is there interesting science at the level even of a whole cell, but now we have many cells in an elastic continuum. Each one is pulling, like I showed you before with that gel that was deformed. Every cell is pulling, and that means this cell feels the pulling of this cell. So just like I showed you before, within the cell, this picture was within one cell, I have actin and myosin, little pieces of molecules that are being pulled. The entire cell acts like a puller and acts with other cells. So we have many, many scales on which this idea of elastic forces can be relevant. All right, I'm going to now tell you a little bit about force dipoles in an elastic medium. and then show you some of the results. So we said that a force dipole are two forces that are equal and opposite, and they're separated by some distance A. And the dipole is defined by the product of their distance and the force. It's an analogy the way electrical dipoles are constructed. Now, I can't teach you theory of elasticity. I define strain and stress, but either in applied mathematics, mechanical engineering, physics, and material science, all those different areas, you're introduced to the theory of elastic materials. A gel is an elastic material. Don't make any mistake about it. You don't have to be a metal or a semiconductor or a standard material to be elastic. Because of these crosslinks, the gel theory of elasticity applies to gels. It's a very weak elastic material compared to a semiconductor or metal. Well, the results of the theory of elasticity say that if I have a dipole, let's say right in the middle here, I have two forces that are equal and opposite, so they're pulling like this, they're contracting the whole medium. Because this is all connected, this dipole is pulling here and it's connected to this gel that's all connected to itself. There's strain in this gel, even far away from the dipole, feels the fact that the dipole is pulling. And the strain decays as 1 over r cubed, where r is the distance from the dipole. So I have a dipole over here, but yet here I have in this particular picture, this dipole, let's say, is compression like you would have in a biological system. Here the system is expanded, here it's compressed, expanded, compressed, and you can calculate exactly how much it's compressed or expanded. It decays away with a very long range. 1 over r cubed is a very long range interaction. It's like the electrical dipole interaction. The symmetry of this dipole is different from an electrical dipole. It's more like an electrical quadrupole, but that's just a technical point. Okay, now there's something interesting about interactions of dipoles. At least, let's do a three-dimensional problem. If I have one dipole interacting with many other dipoles around it, so I just showed you the central dipole, but I didn't show you all these other little pieces of actomyosin. They're all pulling on each other through the elastic medium. So this pulls, the elastic medium gets deformed. This feels the deformation, so they all feel each other. And I want to know what's the interaction energy. I have to integrate the interaction of this dipole with the continuum of dipoles around it. I didn't show you that continuum. They're all smeared out into that orange plot. When I calculate that interaction energy, you have to do this integral. 1 over r in 3 dimensions, r squared dr, 1 over r cubed, an integral over some angular function that I didn't show. Well, something interesting happens. The angular integral vanishes, turns out, when you calculate this. The 1 over r cubed integral for an infinite medium, you see r squared and r cubed cancel, you get the integral dr that diverges. So one part of this integral diverges, the other part of the integral vanishes. What's the value of the integral? Well, it turns out instead of doing the three-dimensional integral, you convert it to a surface integral. And the important point for those of you not interested in integrals is that the interactions of these dipoles care about the shape of the volume containing the dipoles. Now, usually in physical systems, two molecules that interact, they interact usually on a short range. So they might interact if they're 5 angstroms, 10 angstroms, 20 angstroms apart. You don't have to care about, if I'm talking about a metal or semiconductor, I don't usually care about the shape of the metal if I want to know how the molecules or atoms are interacting. But here, because the interactions are long range, the interactions between these molecules care about the overall shape, and they also care about other boundary conditions, namely how rigid is the surface or the environment that this cell is in. So because dipole interactions are long range, in the end, they care about the shape of the container, here the cell that they're in, and they care about the rigidity of the environment that they're in. And here, by thinking about elastic dipoles, we have some hints why you might have cells that care about the rigidity of their matrix. All right, then you can do calculations using the theory of elasticity. And I'm not going to go through these equations. Let's just look what it says in red. Tomorrow's workshop, I'll explain these equations a little bit more. But let's just look at the pictures here in red. You can have a sum of dipoles. Now, this is supposed to represent the cell. And all the dipoles, if I add it up which way they're pointing, the net force they act, they're all inside the cell, but they would act as if there were surface forces compressing. You see, this is equally isotropically compressing the cell. We'll call that time of a dipole P0. There's another kind of dipole that couples to shear, where it might stretch the cell in one direction and compress it in the other. Actually, in cells, the dipoles are also always compressing. But if I have more dipoles oriented in this direction than this direction, relatively speaking, I'll have more compression in this direction, less compression of relative stretch in this direction. And that's a different symmetry of organization of dipoles called P1. The interesting thing you find from the theory of elasticity is that if a cell has P0, if a cell has a tendency to have dipoles in it that will, if they were randomly oriented, would uniformly compress the cell. And we've seen that cells have these actomyosin filaments. The myosin motors exert forces. They exert these dipoles. The cell has these. It makes them. You don't ask why. And let's say they were isotropically randomly distributed with their orientation. The forces would be randomly distributed. There'd just be some isotropic compression. You can show from the theory of elasticity that if you have such a compression, it induces an ordered P1. It induces some of those dipoles, or many of them, to order in the same direction. So once you have dipoles that feel each other, there's a tendency for them to align in the same direction. And the hint for the physical reason of that is shown over here, because if you have one dipole here, you have expansion here, compression here, expansion here, compression here. And another dipole would rather go where the cell is expanded in these red regions rather than where the cell is compressed, because it costs that dipole less energy. The cell, another dipole has to expend less energy. Since it's compressing, it wants to go where the elastic medium was already expanded, because if the medium is expanded and the dipole compresses it, then it costs it less energy to do that, because it's bringing the medium away from an expanded state back towards its equilibrium state. So what you find is that these dipoles interact with the direction. And once you have dipoles that would cause, if they were random, just an isotropic compression, they cause a nematic or orientational ordering. And this orientational ordering depends on the shape of the cell. If the cell were completely round, this does not happen. The coupling between the isotropic and oriented dipoles goes to 0 for a circular cell cross-section. And it also depends on the substrate rigidity due to the long-range interactions. So I haven't shown you the theory, but I've given you a hint from this integral over here that when dipoles interact, they care about the shape, and they also care about other boundary conditions because their interactions are very long-range. And that's why the orientation of these dipoles within a cell care about the overall shape of the cell and care about the rigidity of the substrate. And let me go now to some experimental data where they used some of the theoretical ideas. These are experiments done in stem cells. Stem cells, again, are baby cells. The pieces of actin and myosin are rather small. Should we maybe go to this slide here? This is a picture of these stem cells after 24 hours. The little pictures here, little lines here are pictures of actomyosin. And they're relatively small. They're not all across the cell. They're not very long fibers. And there's an overall orientation to these. In this cell, we measure something called an order parameter. How well are these dipoles oriented relative to the long axis of the cell? So the long axis of the cell is this black line, and the red line is the average experimentally determined direction of orientation of these dipoles. So on a very soft substrate, that orientation, its maximum is 1 when they're all oriented along the black line. So when this angle chi goes to 0, all the little dipoles are pointing the same direction, and S would be 1. This is a very, very low order. On an intermediate substrate, you don't have to know what these units are, but they're different rigidities, we find the most order. And for a cell that is not very far from round, so this cell has an aspect ratio, has a shape, 1.5. That means the length divided, the long axis divided by the short axis is 1.5. For this cell, when you put it on a very rigid substrate, the order parameter went down. For cells that are more elongated, that have higher aspect ratios, the order parameter has a small peak and then saturates as you make the substrate rigid. So the dots here come from some theory that I won't go into in detail, but what's important about this? What's important is that the cells feel two things. these little dipoles, there are little pieces in here of actin and myosin, and you could do experiments at 2 hours and 4 hours where the pieces are even smaller. These little dipoles feel the rigidity of the substrate on which you put them. They feel how rigid is the plastic that you put them on. That's number one. Number two, you see that these curves care about the shape of the cell. This R is just the ratio of C divided by A. It's just the aspect ratio or the elongation of the cell. So the orientation of these little dipoles within the cell is not just short range. It doesn't only care about the distance between the dipoles. It cares about the global shape of the cell and cares about the boundary condition of how rigid the elastic environment you put them in is. And we've calculated this with the theory, and I've given you some hints that just because you have forces in a cell, when the cell gets to be elongated, because these forces are long range, it feels the shape of the cell, and they all tend to align to some degree. S here is very small, S here is bigger. The maximum S you could have is where they're all aligned, but it aligns to some degree along the long axis of the cell because these dipoles feel each other elastically, and those elastic interactions are long range. So we think we here have some physical process in stem cells that cares about the boundary conditions, cares about the rigidity of the substrate, cares about the shape. Now you may say, aha, but at the beginning of your talk you told us the development or differentiation of stem cells cares about the rigidity of the substrate, what proteins they produce, whether they grow up to be muscle cells or brain cells. Is there a connection between the two? And here we can only speculate at this point, although the group of Shiva Shankar that is in the are they still in the Mechanobiology Institute? I think they might be moving to India, but they've been in the Mechanobiology Institute headed by Mike Sheets here on the campus, has done some very nice experiments, and other groups, the group of Ingber have talked about this. What could be the connection between the way a stem cell decides to differentiate, whether it's going to be brain, muscle, or fat, or bone? That cares about how rigid the substrate is. What could be the connection of that and this very physical measurement of an order parameter, orientation, angles, theory of elasticity that also cares about the rigidity of the substrate? Well, when these dipoles align, they are pulling on the whole cell internally. These dipoles are internal to the cell. They're also pulling on the nucleus of the cell. I think everybody in this room knows that the nucleus in the cell is where the genetic fate of the cell is determined. And what the speculation is that what happens is when these dipoles pull in different ways, and obviously if the dipoles are aligned weakly, they'll pull very weakly. Here they're almost isotropic. They're pulling on the nucleus. The nucleus doesn't change shape very much because they're pulling almost in isotropic directions. Here they're pulling along the long axis of the cell. You expect the nucleus might deform much more along the long axis of the cell. Well, if the nucleus deforms through these dipolar forces, then perhaps the DNA packing in the nucleus is modified. And when you change the way, you've changed the shape of the nucleus, you've changed the way the DNA in the nucleus packs, something called Shiva Shankar calls, and other people in biology called chromosome territories. The chromosomes in the nucleus are not just in some liquid soup, but they have a definite organization. And if you have forces like these dipoles that elongate the cell, they pull on the cell and they orient, they elongate the cell, then it's going to also elongate the nucleus, and that will change the way DNA packs. And that ultimately can change the expression of proteins by DNA, can change the genetic fate of the cell. So this is speculation, but it's a combination of something that we understand as sort of physics, that I showed you, these dipoles, elasticity, orientational order parameter, and these experiments on stem cell function. A lot more work has to be done to see if this connection is real, but people are working on this, and real experimentalists, not theorists. Okay, I think I want to finish in about 2 minutes. Let me just tell you about another type of elastic interaction very quickly. Your muscle cells are composed of fibers called myofibrils, and they're also an array of actin and myosin. So the blue here is actin, and the myosin, the pulling molecule, the motor, is shown here in red. And in a muscle cell is, let me go back to this, very well-ordered cell. This cell on the intermediate rigidity substrate is very well ordered. You see that has the highest order parameter of the orientation of these little pieces of actomyosin. That's shown by the peak here. Incidentally, this peaks at a rigidity just where the stem cells made muscles. So the muscles know that their actomyosin has to be very ordered, and that indeed happens, and that's why we think there's a connection between the two. Well, in a muscle cell, the actomyosin is eventually, in a mature muscle cell, is perfectly arranged. So the actin are arranged in these bundles, and the myosin in between them. There are even X-ray diffraction patterns in these things. And the question is, how do muscle cells get organized? And here's a picture of a muscle cell at the beginning of its development, again, work by Disher on stem cells. But these stem cells are further along in their maturity to become muscle cells. So they've already ordered orientationally. Everything I talked about before, it's been done. Those actin polymer fibers are already oriented, all parallel to each other. And here the little red is the myosin pullers. The green are crossed. Now they can be organized in two ways. These are different bundles of actin, what I call stress fibers, that are oriented parallel. We've gone beyond what I showed you in the earlier pictures. It's organized into a muscle. The myosin and the cross-linkers, the green and the red, they could be registered in different bundles randomly, like here. Or they could be arranged in layers where all the myosins in different bundles are together, and all the cross-linkers are also together in the same layer. In the language of liquid crystals, this is called smectic layer ordering, and it's actually found in muscle cells as they develop, that these different bundles of actin, not only are they parallel to each other, but the molecular motors are all in the same layers, and the cross-links are all in the same layers. And the question is that some kind of chemical interaction or not? And what Dennis Disher has found is that this kind of ordering also depends on substrate rigidity. I won't go into the theory, but we've done a similar theory, just show you some pictures from a paper we published jointly with Disher's group in Biophysical Journal on again, stem cells on substrates of different rigidity, but they're going to develop into muscles. It's hard to see here, but here, the bundles of actin and myosin, there's no registry between them. They're like this picture. There are bundles, they're oriented parallel, but the myosin in one bundle has no correlation with the myosin in the others. In the middle picture here, in an intermediate rigidity substrate, the myosins form these layers. It's hard to see because of the resolution of the picture, but what happens on the intermediate rigidity is that you get these layers of myosin, layers of cross-linkers. This is very important to have a muscle cell that all acts together. Obviously here, each bundle is going to act independently, because the pulling molecules, those are the forces your muscles exert when they contract, they're all independent. Here, they're all connected to each other, and this exerts a much more coherent force. And what Disher has discovered experimentally is that that depends on the rigidity of the substrate that you grow this at least one layer of such cells. So this is another phenomenon that I haven't explained to you theoretically, but Benjamin Friedrich, who was a postdoc in my group, used a very similar theory to explain how long-range elastic interactions can also drive this layering interaction between random registry of the pulling motors here and layering registry in here. So I think I'm going to stop here and just put up some things we're thinking of. In tomorrow's talk, those of you attending the workshop, I'm going to review some of this in a little more mathematical detail, but then I'm going to talk about nonlinear elastic effects. The fact that a real actin gel or many biomolecules, linear elasticity means that if you deform it a certain amount, like a spring, you stretch it, the force is proportional to the stretch, Hooke's law. Nonlinear materials, the force is nonlinear in the stretch, and many biomolecules have this property, and there's an interesting mathematical, physical problem. We've been looking at nonlinear elasticity, and that's going to be part of my talk tomorrow at the IMS workshop at 1:30 in Bungalow 2 on, I don't remember the name of the road, St. George's, Prince George's, right. So thank you for your attention. I hope I got across some biology, even though I'm not a biologist, some physics, I am a physicist, and some of the joy of doing theory in an area that's very complex, where sometimes you might have a simple picture that might be useful. Thank you.
The last two pictures you showed us, what was the method you did them? Are they fluorescence images or second harmonic?
As I said, we did the theory.
These images?
No, these.
Yeah.
I would guess they're fluorescence.
Oh.
The paper is in Biophysical Journal 2011. The first author is Friedrich, but you could look for Disher, Safran, or you could email me and I'll be happy to send you a preprint. The paper is in Biophysical Journal. As I said, we did the theory. But I think it's fluorescence.
You mentioned a lot about orientation and some elastic deformation. So is that material have any similarity to pneumatic elastic ball?
Well, the underlying gel, when you have this random actomyosin gel like this, it's similar to sort of a rubber, okay? But what's different about the biological system is you have the underlying gel. You have these myosin motors. And the motors are you don't have this in a dead material. The cell is alive. These motors are pulling on this gel. And that's what's different between an elastomer. It's a very big difference because you have internal forces, small molecular internal forces. Now you say, who's generating the forces? The cell is generating them by burning energy. The molecules consume ATP, and therefore they try to move and they exert forces. So in that, it's very different from an elastomer. It would be like an elastomer if you could put little hands in it that are always with fingers pulling it all over. And that, the cell is very, very different. But there are some material science analogies to elastic inclusions where you'd have an elastic material, and you put another elastic, you drill a hole, and you put another elastic material inside, okay? Rubber and metal and rubber. Okay, everything's all right. You just put a piece of metal in the rubber, you glued it, it sits there. But now, let's say you warm up the system. The metal expands, and then it exerts a force that's trying to expand the rubber. In a sense, that's a little bit like these molecular motors.
So you mentioned this dipole interaction. You have recently in the cold physics, the dipolar condensation. There, apply external magnetic field. Then all the dipole will align in one direction, say fixed direction n, unit vector in R3. Then there also have a dipole interaction. The interaction kernel is like a 1 minus 3 cosine theta square over R3. Theta is the direction between the angle between n and r.
Yeah.
So I'm wondering, the interaction here in your case is similar to that case or different?
It is very similar to that case, but what you defined was how you describe the orientation of the dipoles. I'm talking about the energy of interaction in a usual liquid crystal that might be aligned by a magnetic field or an electric field like in your watch. The dipoles usually interact only in a short range. But here, because these dipoles are pulling on the elastic gel, they interact with a very long range. So in that sense, it's different. The second question I think you're asking is, can you use an external field to align these? Here, the experiments I showed you where they align, they do it spontaneously. You just put them on the right substrate of the right stiffness, and they will start to align spontaneously because of their elastic interactions. What aligns them here, the analogy of the alignment, is the cell shape. Remember, I told you, if the cell is round, they don't align. There's no cue that tells them which direction to align. Here, they're aligning along the long axis of the cell. The boundary condition of the cell shape, which also comes into the dipolar interaction because it's long range, so it feels the boundary. That boundary condition in the cell shape is like a field that aligns them. And that's why the different shaped cells, this R is the ratio of the long axis, short axis, different shaped cells have different data over here. So that's like the field. You could also put a stretching field on and a curvature field in this.
So basically here, the different dipole, the direction may be different.
It's not short range. Usually for liquid crystal molecules, those molecules feel each other over a nanometer. And here they feel each other over a very long range. And that's why they're sensitive to downward motion.
The initial example which you showed of a stretching membrane and the cells aligning perpendicular to it, right? I assume the fibers are stretched in the vertical direction,
Yeah.
right? That looks very exactly opposite to how muscle cells work, where the fibers are along the stretching direction.
Right, but this is stretching where it's dynamically stretched at 1 hertz. So the muscle cells are contracting macroscopically much slower than that, and it's a different situation because you're stretching it at a high frequency. And what we think theoretically happens there is that when you stretch the substrate and the cell like so fast, the dipoles cannot, they don't have time to orient to the direction. So they say, if I would orient, I'd be very frustrated because you're always changing the magnitude of the strain, and then they're going to have to adapt, and they can't adapt. And so they go perpendicular. They actually go to the zero strain direction, and they go to a direction they don't feel any stress because they're too frustrated by the dynamics. It's really a dynamical problem.
Okay.
That's a good question.
Thank you.
Well, Professor Safran, I entertain your talk a lot. I still have a question for your comment, but I'm afraid that I'm not able to express my question very properly.
I might not know the answer.
Well, your talk gives me a very clear sense in the soft matter physics. While, according to my understanding, the function of the cell is very robust. It is kind of structured to perform a very particular function. And so how do you reconcile the very general principles applied to soft matter physics and some quite unique function that is inherent in a biological cell?
It's a good question, and you can study even the well-adapted and mature cells. You can study parts of that with this. But if you notice, I stressed that the applications we were talking about mostly were stem cells. Stem cells are cells that are not robust. They're cells that can develop into any kind of cell. And you change a little bit of the biochemistry. You change, as I showed at the very beginning of the talk, remind you of that. You change just the rigidity of the substrate you put them on, and the stem cells grow up to be completely different kinds of cells. And they express different proteins. So I think you hit the nail on the head, actually, because the best application of these sort of more wild ideas where you start with the interactions and they can do anything, and then you try to predict what they'll do. In a mature cell, indeed, there are many, many, many feedback loops and biochemical loops that we can't put into this. In the stem cells, it seems that these loops are much less well established. And perhaps because stem cells are not robust, because they can become anything, maybe that's what applies. I think you've asked a very deep question. I'm not sure my answer is as good as your question.
Any more question?
No, just a little. I mean, I'm still not sure about this increasing substrate rigidity would really cause a stem cell to change its shape so
Well, I'm
much
not sure either.
in natural environment, for example, in a biological tissue. One in biological tissue, other than bone, there is no hard or rigid substance for a stem cell, for example.
Oh, but okay, so there are two questions. One is understanding these experiments where they put the stem cells on plastic.
Yeah.
Okay, and that's sort of done by a group that's coming from soft matter physics and have moved into biology, the Disher group. And we want to understand this also because it's important for the processing of cells. And the other one is, where is it relevant? I'll tell you something interesting in one of Disher's papers that he quotes. If you put heart muscle cells, stem cells, that you put the right biochemistry, and you put them in a blood vessel, which has had an infarction, so where the blood vessel is damaged, right? And one of the obvious things you'd want to do is put stem cells there, and the stem cells should become blood vessel cells and heal the damage. That's what you would think. What happened was they developed into bones. And the reason they did is because the damaged area is very, very rigid elastically. And because it's rigid elastically, the stem cells got a cue that they should go to the bone. And it's therefore using stem cells for the body to heal itself is a more complicated process. And I'm just quoting what's in one of Disher's papers, and they talk about this. So there are parts in the body where there are damages, and the rigidity changes from what the rigidity should have been. And these rigidities are rather soft. These are the rigidities of these cells in this scale. They're kilopascals. A piece of glass is many gigapascals, so orders of magnitude softer. But this very dramatic example, I don't know if they tried it on a person or an animal, but of trying to heal heart tissue that's damaged, exactly point out here.
Maybe I'll ask you a question. See, when we exercise, right, we put stress on our cells, right? And we know that when we do exercise, our muscles build up. We build up our muscles through exercises. How much this theory can explain that?
We actually, there's probably a relation, but we didn't do that. But we have a young biologist, Elie Zeltzer at the Weizmann Institute, who studies development in early embryos, and he studies the fact that bones develop correctly only if the muscles are pulling on the bones. So you know your muscles and bones are connected by ligaments, and if the bones are trying to develop and the muscles are disconnected from them, bones don't develop correctly. And I know that when people have osteoporosis, have a weakening of their bone structure, they're told to do weight-bearing exercise because the muscles pulling on the bones somehow gives them some cue to develop differently, and that's all I really know. But
That's very fascinating,
yeah,
very interesting.
Yeah, I mean, I'm sure that there are studies by development.
Any more questions? If not, I'd like to present a little gift to Professor Safran as a small token of appreciation for your very wonderful lecture. Well, so let's please join me again in thanking Professor Safran for his wonderful lecture.